In this video, we're going to introduce nerve cells. We'll start off by talking about neurons, then we'll talk about glia. Neurons and glia together will be the neuroglial cells of the nervous system. So when you hear nerve cell, that's what we're referring to. Most commonly, people are going to be referring to neurons, but glia are nerve cells as well. There's more in our brain than just nerve cells. We, of course, have epithelial cells, and among those we have the vascular cells as well. They're going to create the blood-brain barrier and will importantly supply blood to the brain. And we'll end by talking just a bit about how nervous systems have changed throughout evolution, kind of at a macro level, and then we'll funnel it down, of course, to the micro level. This is molecular neuroscience after all. So neurons are, I suppose, typically the star of the show. They're excitable, they're polarized, and they are well equipped to communicate over long distances in a very brief period of time. So neurons are all about communicating. An active neuron is a neuron that's communicating. We're going to walk through very quickly the morphology of neurons. You're very much familiar with this, so I apologize in advance, but we have to. So we have our cell body right in the center. This is exactly what it sounds like. This is essentially the cell. Everything that you'd find in a normal cell is going to be found here. Most importantly, we have protein synthesis going on. There is a little bit of protein synthesis in the dendrites as well. We'll go ahead and add a little branch point there. And why not? We'll add another set there. Wherever the soma lives, that's where we say the neuron lives. For example, your sensory neurons that innervate your skin, joints, muscles, those live in the dorsal root ganglia because that's where their cell body is, even though their dendrites and axons are out there in the periphery. So the cell body is where the cell lives. Because if you don't have protein synthesis, you're not alive. The dendrites are going to, of course, act as the receptive end, except when they don't. But we should think of them as that. We'll think of them as the ears. They can also release neurotransmitters as well. But we'll tend to think of the dendrites as the receptive end, and information is going to flow from them into the cell body, eventually reaching the axon. And we'll think of the axon as the mouth. This is where the neuron speaks. Of course, it also has synapses on it as well, so it can receive input. Those are going to modulate the degree to which this axon communicates with its partner. But this is a pretty decent simplification of the flow of information. Input tends to come in at the dendrites simply because they have a lot more surface area than the cell body, although the cell body can also receive synaptic input. That input from the dendrites flows into the cell body, and if we're lucky enough to kick off an action potential, we fire that down the axon. Dendrites are tapering. That means they're going to get smaller as you move away from the cell body. What this leads to is an increase in internal resistance, and that's going to affect how charge flows. So this little simplification does matter. The tapering of dendrites is going to affect how electricity flows within them. We'll talk more about that in Lecture 4. Now, we say that neurons are polarized not just because they have a charge across their membrane, but because they have these two ends, a receptive dendritic end and a transmissive axonal end. The axon is going to be uniform in diameter. It's also going to actively propagate signals down its length. Dendrites can do that too. The axon is usually going to be the site of synapse formation. Now, we usually draw the axon as a nice straight line down to its targets. You need to keep in mind that the axon can branch as well. So in this neuron, we can see a little stellate dendritic arbor there. cell body in the center. The axon comes off, it enters the nucleus, that it innervates, and then it spreads out to communicate with many cells. And that synapse is going to look something like this. So at the synapse we have a presynaptic site, we have a postsynaptic site, and you can tell them apart very simply by looking for the vesicles. So oftentimes we're going to draw a whole bunch of space between these. There's really almost no space, 20 nanometers or so. But you're going to look for the vesicles, those little packets of neurotransmitter that you're already well familiar with. That's going to tell us presynaptic, this postsynaptic density that has all the receptors. That's going to show us postsynaptic. You know I won't sleep tonight if I don't label this. Okay. Postsynaptic tends to be a dendrite just because of their surface area. But it could be anything. Axons can form synapses on dendrites, cell bodies, and other axons. But here's our general central dogma of nerve cell signaling. Input at the dendrites, funnel into the soma, down the axon, spit out some neurotransmitter at the synapse, which would be onto another cell's dendrites. The basic function of the soma is to be a cell and to stay alive and to create proteins. That's the big job of the cell body, and there's a couple reasons why it's well set up for that. First of all, we have a nucleus. Without a nucleus, we have no DNA, no RNA, and thus we can't make proteins. The other important substance would be the Nissl substance. So this is showing you a Nissl stain. So the degree of purple is going to show you not school pride, but instead the presence of ribosomes. So the more purple you see, the more ribosomes you see. And what you'll notice in this real live neuron is that the cell body and the dendrites are purple. You see a lot more purple in the cell body, but there's still some purple out in the dendrites. So we can synthesize protein, certainly in the soma, a little bit in the dendrites, but you'll notice that the axon is devoid of Nissl substance. No ribosomes, no protein production. But of course we need proteins to do everything in life, so the cell body is the site of synthesis, and then they're going to be transported down the axon. In the cell body we'll also see summing of electrical potentials from the dendrites to determine should we fire an action potential or not. But the primary function of the soma, at least the active function, is going to be protein synthesis. So in this case we have two dendrites coming off. This would be a multipolar neuron. If it just had the one here, it'd be bipolar. The number of dendrites that comes off of the cell body is going to be variable. Humans won't have any, I don't think humans have any true unipolar neurons. I'm going to walk it back a little bit. I believe all of our unipolar neurons are actually pseudo-unipolar. I think vertebrates always start off as bipolar and then they fuse their dendrite and axon together. So not every neuron is going to have a true dendrite that's coming off of its cell body. So if you were to look out in the dorsal root ganglion, you'd see the cell body. There'd be that axon coming off. It's going to go back into the spinal cord, microspinal cord right here. And then the axon will extend out into the periphery and create some dendrites. There's no dendrite coming off of the cell body. So this is one of the variable features. How many dendrites do they have? How are they arranged too? So if you look in this illustration on the left, you'll see pi, py, that's labeling a pyramidal neuron. So they have kind of this triangular cell body with an apical and then basal dendrites. Others have more of a star shape, where there's dendrites radiating off all of them. We call those stellate. This can help us distinguish different types of neurons from one another. Those large upper motor neurons that live in our motor cortex, those are going to be pyramidal. but the more local circuit neurons tend to be stellate. Another way that dendrites can be different is the presence and absence of dendritic spines. So coming off of dendrites, we can have little spiny protrusions, and these are going to be the sites of excitatory synapses. I believe inhibitory synapses do also form on the neck, but forget about that. When you hear dendritic spine, just think excitatory synapse. And the reason that these are so cool is that they can grow or shrink without affecting the rest of the dendrite. So these are very plastic sites for synapse formation to occur. We can build new ones, we can get rid of them, and all the rest of the dendrite and all the other synapses remain intact. Pretty nifty stuff. They also have very high internal resistance, which makes them wonderful postsynaptic targets. I think that's going to be lecture 4 as well. Not lecture 4 would be the action potential. That's going to be lecture 3. The action potential is what's going to be transmitted down the axon. Now, the axon is well suited for generating action potentials because of the high density of sodium channels that we find in the axon, particularly at the initial segment of the axon. Sometimes you'll see it written as the axon initial segment. So if you ever see this, that's what it's talking about, the axon initial segment. So that very first part of it has a high concentration of sodium channels. It's about 45 times higher than you're going to find in the dendrites. So we need a lot of sodium channels, of course, to generate action potentials. So if you're going to generate an action potential, it makes sense to do that somewhere where you have a shitload of sodium channels. And that's what we find in the axon. If you look at the data here, red is showing you sulfurrhodamine. So look at that neuron on top. What they did was record the activity. So they did patch clamp recording, but they had a little dye floating around in their pipette internal solution. So when they broke open the hole, that dye, called sulfurrhodamine, filled up the neuron, so you can see it red there. They then took that and stained it with an antibody against sodium channels. That's the PAN, just means it's going to hit a whole bunch of them. NAV, voltage gated sodium channels. And lo and behold, you find them in the initial segment of the axon. So we got a nice, let's see, bright red cell body, and then coming off of that, I'm trying to mirror image this, is that green extension. That's showing you the initial segment of the axon, and below that, they're just showing you sodium recordings. They used a dye that lights up when sodium concentrations increase, delivered through their pipette a series of action potentials, and saw as the level of sodium built up and then dissipated over time. This is all a very long-winded way of saying action potentials use sodium to cause depolarization. I'm sure you've heard that dozens of times by now. We'll walk through this again later on in lecture 3, going through the different parts of the action potential, the dipole, repole, after hyperpolarization, all those steps. Now sometimes the axon can be myelinated. This myelin is going to affect the electrical properties of the axon, and what it will do is speed up action potential propagation. Pretty nifty stuff. The main thing that it's going to do is drop membrane capacitance to allow for more rapid transfer of charge so it doesn't get held up in the membrane. Some axons are myelinated, some aren't. Those that travel long distances and need to do so very quickly are going to be myelinated. So, motor axons, so that we can move our limbs. Sensory axons, so we can feel tactile sensation. The painful ones, some are myelinated, some aren't. The whole point of firing that action potential down the axon is, of course, to cause the release of neurotransmitters. So now we've completed that central dogma of nerve cell signaling. We have electrical and chemical. Electrical within, chemical between. The electrical within starts at the dendrites, goes into the cell body, then hits the axon to generate the action potential. Something every neurobiologist should be well aware of. That action potential travels down the axon, we spit out neurotransmitter onto the dendrites of another neuron. There's our synapse. That's going to generate electrical activity in those dendrites. It's going to tend to flow toward the cell body because of that higher internal resistance out there away from it. That's why it's important that dendrites taper. They're going to follow the path of least resistance into the cell body. Rinse and repeat. Now the type of neurotransmitter that we spit out is going to matter. Some neurotransmitters are going to be inhibitory. Almost all the time. GABA is, at least when you're an adult, GABA is inhibitory. When we're growing up, not so much. But we should think of GABA as an inhibitory neurotransmitter. It's going to decrease the likelihood that we fire an action potential. It's going to largely do that through shunting inhibition, which we'll talk about in, I believe, Lecture 4. Might be 2. I'm pretty sure it's 4, though. I probably should have looked. Alright, have a look at the data in this slide. So on top, what we're looking at is a voltage ramp. So that black line that's kind of ramping up shows 100 picoamps. They're applying 100 picoamps of current over time. And the purple trace is showing you the spiking of a dopaminergic neuron. So when they increase the amount of current that they deliver it, when they stimulate it electrically, it fires action potentials at a much higher rate. Now look at this same voltage ramp on the bottom. That blue bar is showing you where they shine blue light onto the slice, and that blue light stimulated GABAergic input onto the dopamine neurons. Notice the difference in the frequency of action potentials. Now we all are familiar with the concept that action potentials are all or none, and frequency encoding determines how strongly a neuron communicates with its target. Frequency drops, and so the output of the dopamine neuron is, of course, weaker as a result. These GABAergic neurons that inhibit other neurons tend to be stellate, and they're usually aspiny. A notable exception would be spiny projection neurons in the striatum, which are spiny, but they project outward. So the projection neurons can make an exception to that. And the inhibitory neurons can be local circuit, or they can be projection. You tend to find them more as local circuit, providing more local inhibition to create logic within an area of the brain. Their synapses are going to tend to occur more in the proximal regions of the dendrites and on the cell body. This is what's going to allow them to be much stronger. So we're going to see our inhibitory synapses on the more proximal dendrites, and further away is where we're going to tend to see our excitatory synapses. We will talk about why in Lecture 4. So we have inhibitory neurons. Of course, we must also have excitatory neurons, which you've already heard about, I'm sure, by the time you're in this class. When we're thinking excitatory neurotransmitters, we should be thinking glutamate for the central nervous system, acetylcholine for the peripheral nervous system. Of course, with inhibition, we think GABA, glycine for the cerebrum and brainstem. But we're moving on. We're on to excitatory neurons now. So look at the data on this slide. The blue line again is showing you where blue light was used to excite neurons. So in this case, what we did was express channelrhodopsin under the synapsin promoter. Synapsin is just a protein that you're going to find at synapses, so every neuron is going to express it. So we put channelrhodopsin into neurons, and these were excitatory neurons. We then hit blue light to have them spit out their excitatory neurotransmitter, and if you look at the action potentials here, these were recorded cell attached, so they're just going to be vertical lines. You see a much higher frequency of vertical lines, thus action potentials, while the blue light is on, while glutamate is being spat out. So the purpose of glutamate is to increase the frequency of action potentials. The purpose of GABA is to decrease the frequency of action potentials. That's what you should be thinking of when you think about their function. What's their impact on the frequency of action potentials? Don't think of inhibitory neurotransmitters as being there to hyperpolarize a neuron. That doesn't do shit. The purpose of a neuron is to communicate. If it's resting or hyperpolarized, it's probably spitting out just as much neurotransmitter, so there's no effect. That's not the purpose of inhibitory neurotransmitters. The purpose of them is to slow down the frequency of action potentials or to prevent the initiation of action potentials. We don't really care about hyperpolarizing neurons. It doesn't do anything. Because neurons aren't there so that we can measure their membrane potential. They are there so they can communicate with their targets. So, excitatory neurotransmitter increases the frequency of action potentials, increases the amount of neurotransmitter that neuron spits out. You're going to see more often here spiny neurons compared to inhibitory neurons, but not all of them are going to be spiny. You see different types of morphologies. They could be pyramidal. They can also be stellate. The pyramidal neurons are going to be excitatory, though. They can be local circuit. They can be projection. And like I said, the site of their synapses are going to be more distal. So that excitation has to pass through an area that can be inhibited. And this allows inhibition to trump excitation. And that's a much safer way of setting things up. This way neurons won't be won't go through excitotoxicity. They won't be excited to the point that they die. And we'll talk about why this matters later on in Lecture 4. So we have inhibitory, we have excitatory, and then we have neuromodulatory. And these can be excitatory or inhibitory. It depends on the receptor that they act on, but the big difference here is that they act over very long time courses. So these essentially set the state of a neuron over a long period of time. They're not going to cause instantaneous and immediate changes in the firing rates. So if we walk back one, so stimulate an excitatory neuron, you get an immediate increase in firing rates. Remove the glutamate, you get an immediate return right back to where it was. So glutamate and GABA are going to act, for the most part, over very short time periods. That's absolutely true if they're acting on their ionotropic receptors. There are some metabotropic exceptions later on. Of course, there has to be exceptions to everything. The neuromodulatory transmitters don't really have that ionotropic component, except for 5-HT3 serotonin receptors, but all the others are going to be metabotropic and thus act over a much longer time period. So with neuromodulatory transmitters, we're going to have long-lived changes in the excitability of a neuron. So when we're thinking of neuromodulatory transmitters, we should be thinking about things like our catecholamines, so dopamine, norepinephrine, there's some epinephrine as well, and also serotonin. Acetylcholine could also be put in here as well. And these neurons are going to be projection neurons, just as a rule, because they live in very specific nuclei in the brainstem. For example, your dopamine neurons, you're going to find them in the substantia nigra, pars compacta, and the ventral tegmental area. Serotonergic neurons are going to be found in the raphe. Noradrenergic in the locus coeruleus. So they have these very discrete populations down in the brainstem. You can see them in this cartoon. How in the world are they then going to serve the rest of the brain? Well, of course they have to extend their axon into those regions. So they're going to have very long axons. They're going to be widely distributed. They're going to serve a large area of the brain as a result. And they're going to serve the brain with a fairly constant release of neurotransmitter. So most of these neurons are going to pacemake. And that's exactly what it sounds like. They're going to constantly fire action potentials at a fairly steady pace. Now the reason for this has to do with the flexibility of communication. Let's say that our dopamine neurons didn't pacemake. So they are at rest, giving us, this will be our dopamine input, they're going to give us 0, whatever that is, and this is going to be max. So it's pretty easy to imagine how we'd get an increase to say, let's say that we enjoy something. So we get reward. There's going to be a release of dopamine. Or we think something's a good idea. Yes, I should carry out that action. So we get a release of dopamine when something's a good idea, and then we're back down to rest. What if something's a bad idea? How do we ever say that there's less dopamine around if we're not releasing it at baseline? So this doesn't work. This can only tell you when things are good. But what about the difference between neutral and bad? You can't distinguish them. So instead, rather than having zero dopamine release at baseline, what we see is what's going on the slide right there. This is a dopamine neuron in the ventral tegmental area. And they're just recording action potentials from it. They just put a pipette on it and didn't do anything. And indeed, if you apply tetrodotoxin, if you block the glutamatergic receptors using, oh my goodness, I'm blanking, NBQX and AP5, for example, your AMPA and NMDA receptors. So you block excitatory input, you can block all neurotransmitter receptors, they're still going to fire action potentials. They have these intrinsic mechanisms of generating action potentials that don't rely on input from the dendrites. So there's an exception to that central dogma we just went through. So they're always pacemaking somewhere between min and max. So if something's a good idea, sure, I'll give you an increase. What if it's a bad idea? I'll give you a decrease. So here we're more likely to do it. And here we're less likely. We can actually have two different directions of communication because of this pacemaking. And so these neuromodulatory transmitters are going to act over long periods of time to establish a state of neuron excitability. So for example, with decision making, if we're trying to decide should I do something or not, I think a great example here is cocaine use. If you see anyone who's intoxicated with cocaine, they tend to be very animated, they talk a lot. There's a lot of output. Every motor plan is a good idea, and they are more likely to carry out all of them because the amount of dopamine floating around in their dorsal striatum is much higher than normal. Because cocaine is going to inhibit dopamine reuptake, there's more dopamine floating around, everything seems like a good idea. Alrighty. And then there's a picture of a synapse. Alright, we'll get to this later. The idea there is that we have a constant level of neurotransmitter floating around in the synapse, so we can have bidirectional communication. Okay, the synapse that we're going to talk about is really chemical synapses. We should keep in mind that electrical synapses do exist. These are gap junctions where two cells share their cytoplasm. These tend to occur through the dendrites, again because they have much larger surface area. This is where you're going to see most of the gap junctions in neurons. But this will allow neurons to communicate with one another immediately and synchronize their activity. Some neurons need to act as a unit to provide synchronous output, and gap junctions will let them do that. But when you hear synapse, just think of chemical synapses. That's the most common type. And of course we're going to have a presynaptic site and a postsynaptic site. The presynaptic site is where we release neurotransmitters. The postsynaptic site is where we receive those neurotransmitters on some kind of receptor. And everything that we talk about at the molecular level, it's all a probability. So when we send that action potential down the axon, that increases the probability that voltage-gated calcium channels open up. Not all of them do, but most do. And that calcium will float around. Some of it will bump into calcium sensor proteins and change their function. And some of those are going to then cause the vesicle to fuse, and the neurotransmitter will diffuse out, and some of it will bump into a receptor, some of it won't. So there's a lot of chance involved when we're talking about how the world functions on the molecular level. But in general, these chemical synapses are going to translate an action potential into neurotransmitter release, and that neurotransmitter into an electrical potential in the dendrites of the target neuron. Now, of course, these are not floating around in space like this cartoon suggests. It's more like this cartoon suggests, where there's cell adhesion molecules that hold our pre- and postsynaptic sites together. Now, whether you're excitatory or inhibitory, there's going to be a presynaptic site, there's going to be a postsynaptic site. They're going to be about 20 nanometers away from one another, and they'll have a diameter somewhere in the ballpark of 300 nanometers. There's a difference, though, in the types of cell adhesion molecules that are there and the types of proteins in the postsynaptic site, so that the excitatory synapses tend to have kind of a thickening of the postsynaptic site because of the adhesion molecules there. And so we call these asymmetric synapses because the postsynaptic membrane is a little bit thicker than the presynaptic membrane. With inhibitory synapses, because they have different receptors and different scaffolding proteins, they don't have this swelling, and it's a little more symmetric. Of course, location will also help tune you in as to whether it's excitatory or inhibitory. Excitatory tends to be more distal, inhibitory tends to be more proximal. Of course, it's really hard to tell that when you're looking at an electron micrograph like this one here. They all look like a bunch of nothing to me. But apparently we have a symmetric synapse on top, that would be inhibitory, asymmetric on the bottom. Let's move on to the glia. We're going to be talking about the myelinating glia, which myelinate except when they don't. There's myelinating and non-myelinating oligodendrocytes and Schwann cells. Then there's astrocytes. These are going to do a whole bunch of different things. I like to think of them as the mother of neurons. And then microglia, which are going to be the immune system. So we'll simplify oligodendrocytes and Schwann cells, and we'll say that they synthesize myelin. That's totally true. They do some other things, but that's a really important function, so we'll focus on that. Myelin is going to, of course, wrap around the axon and ensheath it in this fatty insulation. The problem that myelin tries to solve is, how can I conduct signals more rapidly? Now, you and I, we're both vertebrates, so we can talk about this. Those invertebrates are solving the problem the wrong way. Now, they don't have to worry about a skull. They don't have to worry about all the complex thoughts that we do. So they have fairly simple nervous systems, and the way that they increase the rate of action potential propagation is to just add more lanes. Is this axon not thick enough for you? Okay, fine. Widen that bitch out. The internal resistance is much lower here than here. Because we have lower resistance, that means that our current flow is going to increase. Remember, V equals IR. You're going to hear this a few times. So the current is going to be, of course, driven by that potential difference, but also decreased by resistance. So decrease your resistance, increase current flow. That's what invertebrates are going to do. If we did this, our spinal cords would have to be the size of a sequoia at Yosemite Park. But we don't do that. We don't just widen it up. This is what invertebrates do. It doesn't work well whenever you have the confinements of a skull and a spinal column. So, the skull, the vertebrae, they confine the space that we can work in. So what we do instead is myelinate. So by wrapping multiple layers of myelin around the axon, we're going to separate the charge that's inside from that that's outside. We're also going to prevent leakage of charge from the outside, and what these are going to do is allow for rapid propagation of action potentials from one node to the next. We call this saltatory conduction. Saltatory meaning to leap. I believe saltaire is the word that's derived from. So the myelin sheath is going to create the internodes, or the region between nodes. And at the nodes, sometimes you'll hear them called nodes of Ranvier. These nodes are where we have ion channels. So this is where we will restore the action potential amplitude back to its max, create a fresh round of depolarization, which will then spread passively in both directions, but we'll have a net forward direction because of sodium channel inactivation. In the central nervous system, oligodendrocytes are going to myelinate multiple axon segments, and they're going to do so with the compact form of myelin because, well, we got a skull, so we don't need as much cushioning. We also have vertebrae as well. We don't need as much cushioning because we have that bony protection. In the peripheral nervous system, where we don't have bony protection of our nerves, we have Schwann cells. And Schwann cells are going to myelinate one internode, but it will be a much fluffier form of myelin so that we have protection against injury. Schwann cells are going to be permissive to axon regrowth, whereas oligodendrocytes prevent axon regrowth. And this is one of the reasons why we have differences in the ability for the peripheral and central nervous system to recover from injury. So spinal cord injury, you can have some degree of recovery, but not a whole lot. You sever a peripheral nerve, you can get full recovery depending on how much damage there is. That's because Schwann cells allow axons to regrow, but the logic of our central nervous system is that once we've established this kind of complicated pattern of connectivity, let's not have any big rearrangements anymore. So the oligodendrocytes have proteins that prevent axon migration. Alrighty, so moving on to astrocytes. These would be star cells, and I think as far as the glia go, they are the star of the show. They do a whole lot of different stuff for us. And as we study them more, we find out that they do additional things. So during development, they can help track axons from one location to another, so they'll provide guidance cues. They can be the physical road that migrating cells move along, like a radioglial cell, for example. Depending on what they release, they can help create higher densities of synapses, for example, so they can help with synapse formation. They'll help maintain synapses even after they're formed. So this cartoon here is showing us now in green the glial component of a synapse. So we've got a presynaptic site on the left, postsynaptic, kind of that baby blue right there in the middle. What happens to the neurotransmitters that we spit out? So let's imagine we've got ourselves a synapse, and this is how I pretty much draw them all the time. We release our neurotransmitter. What happens if we don't clean it up? Well, it's going to hang around and it's going to continue to stimulate that receptor, or maybe it's going to diffuse away. And actually we have extrasynaptic receptors, but these tend to be toxic, because that tells us something is wrong. God damn it. Something is definitely wrong. Okay, now it's right. So these extrasynaptic receptors are telling us there's something wrong with this synapse. This communication is no longer specific for this synapse. It's leaky. We should probably get rid of it. It's not working right. To prevent this, we have astrocytes that will ensheath this. I wish I could draw it in 3D, but just imagine this is a hand, kind of wrapping around it there, and it's connected to a cell somewhere out there in the board. As that neurotransmitter tries to diffuse away, the astrocyte picks it up, so we don't stimulate extrasynaptic receptors, no toxicity. And these are fairly important, it turns out. So if we were to get rid of the glutamate transmitters in astrocytes, for example, well, what we get is restricted growth. So if you look in panel A, that's the body weight. And we have our open circles, that's just your run-of-the-mill wild-type mouse. Filled circles are showing you a glutamate transporter knockout mouse. This would be a glial-specific glutamate transporter, excitatory amino acid transporter 2. In panel B, we're looking at survival. So most mice should survive in the 14 weeks that we're plotting here, or maybe 13. But you'll notice that that filled curve is dropping. So the mice are dying off, and you can see why in panel C. They're having seizures. They're having intractable seizures. You can see the recordings of from their EEG that's showing the epileptiform activity in panel D. So because we're not picking up this glutamate, it's going to lead to excitotoxicity, not only for the neurons but the organism itself. In addition to cleaning up neurotransmitters, astrocytes will also help clean up ions. So for example, whenever we fire an action potential, we bring in sodium, we spit out potassium. It's fairly important to buffer extracellular potassium levels, which we'll see in Lecture 2. So astrocytes are going to contact neurons in a number of locations. They'll do so at synapses. We call those perisynaptic processes. They also have perinodal processes. They contact the nodes of Ranvier to help pick up that potassium from an action potential, for example. They also have infoot processes that are going to terminate around blood vessels. So they're going to stick these infoot processes up to the blood vessels, and it's going to cover essentially all of the vasculature within our central nervous system. We're going to find an astrocyte there. And what it's going to do is help maintain the blood-brain barrier so we don't get a bunch of toxic stuff just freely floating into our brain. It's also going to help pick up good stuff that we want. So let's look at this figure and make sure we know what's going on here. So CX30, that's just a connexin protein that helps form intercellular junctions between cells. So astrocytes have it. It helps form gap junctions. The thing that we care about here is the white, GFAP. GFAP is an intermediate filament that's found in mostly astrocytes. It stands for glial fibrillary acidic protein, and so it gives you a rough idea of astrocyte morphology, and we can see these astrocytes are clearly surrounding a blood vessel that's running through the image. Now GFAP doesn't show you all of the astrocyte. We've got to keep that in mind. This is just an intermediate filament that doesn't occupy all of the space. It gives you a rough idea of where an astrocyte is, but if we compare the GFAP signal in red to a biocytin fill in green, so in this case what they did was fill this astrocyte up with biocytin. They can then use an antibody to find biocytin, which shouldn't be in the little mouse brain, and it wasn't. It's only in that filled astrocyte. You can see the green and the red agree for the most part. I mean, they're in basically the same place, but there's a lot more green than there is red there. So there's a lot of astrocyte morphology that you don't appreciate if you just look at GFAP. But because they're filling up essentially all the space around them, what that lets them do is buffer all the space around them to make sure that neurotransmitters don't build up and that there's no dramatic change in ion concentration. Also, this allows them to communicate with one another so that they can help one another buffer neurotransmitter and ion levels. So let's say that this synapse is really, really active and we start to have an increase in our glutamate concentrations here. Well, luckily, this astrocyte is connected to its neighbor. It's somewhere out of the board. Sorry, we need a new cartoon for this. Alrighty. Pre, post. I have to do this. I apologize. Okay, so we spit out our neurotransmitter. This astrocyte here is the lucky winner, and we're going to have it send out a few processes. It's pretty hideous. I'll give you that. And then here's our network of other astrocytes that are all connected. God damn it. This is awful. This is just plain awful. Okay. You know, we're going to draw them like this, like almost like little stars, or I guess diamonds. And now that they have a nucleus, we can accept that they are cells. Okay, cool. Believe it or not, this is our astrocyte network. So when we start to pick up all that glutamate, rather than just building up in this cell, it can spread the love. So that keeps glutamate concentrations lower here, so it can continue to pick it up out of the synapse effectively. It takes a village. I don't actually know what that means, but I always say it whenever I bring up astrocyte networks. I should probably look that up someday. So, by working together, astrocytes can more effectively carry out their function, and that is take care of neurons. I like to think of these as the mother. They clean up everything. They also provide nutrition to the neuron. And the neuron is just there making a mess. It's just crying all day, just communicating, just spitting out neurotransmitter. And if we don't clean it up, it'll kill the organism. We just saw that, I think, 2 slides ago, something like that. And there's a last kind of mysterious function of astrocytes, and I hope that this movie records properly. Sometimes it doesn't. If it's going well, you're seeing some flashing here right now, and if it's not going well, then there's probably just some arrows and nothing going on. Anyway, we can look at this together, but this movie I took from a brain slice from a very special kind of mouse that has it has a type of green fluorescent protein that only gets put together when there's an increase in intracellular calcium. So you get these flashes of light that will show you when there's increases in calcium. And so here you can see a few different cells having little flashes of calcium, and it seems to kind of, in some cases, pass between cells. So rather than firing action potentials, astrocytes create calcium waves. And we think it probably has something to do with communication with one another or with neurons, but they're still pretty mysterious. So astrocytes are kind of excitable, but not like neurons, not where they have these action potentials. Instead, they seem to excite calcium waves. Kind of a mystery. The last glial cell that we'll talk about here would be the microglia. We could have mentioned ependymal cells. They make CSF. There you go, they were mentioned. Microglia are the immune cells of the brain. So if you had to think of one function for them, immune function. Think of them as macrophages. They are more than that, though. They also release signaling molecules. They can affect how neurons work. They can affect development of glial cells, formation of blood vessels. So they do a little more than that, but their main function is to lie in wait. That's what we see in panel A on the left. In panel C, this is post-injury. You can see they've changed their morphology now. They're not so complicated. They look like little neurons there in A, don't they? And that complex morphology makes it makes it difficult to move around. This is why neurons are post-mitotic, meaning they don't go through the cell cycle. How in the hell do you cut this in half? You don't. You kill it if you try to go through the cell cycle. Now, microglia don't have the axon, but they do have these little extensions coming off. So they're just sitting there listening to what's going on, and when they sense some sort of damage, usually it's going to be a change of pH, it's traumatic, or some inflammatory signaling, they then go from this complex morphology to something that's a little more amoeboid, so it's easier to crawl around. They will migrate to the site of injury, clean up whatever's there, whether it be an invader, so some sort of pathogen that has made its way into our body, or just damaged cells. They'll clean it up. So we got our neuroglia down. We should also think of vasculature. We got to think of the vascular cells that are there. So blood is going to have our glucose and our oxygen to help keep our brain fueled. It's also going to have some nasty stuff in it. Sometimes by accident, sometimes we plan it that way. And we want to keep that out of the brain. The way that we do that is through the blood-brain barrier. So neurons, like I just said, are post-mitotic. They're irreplaceable. So we want to make sure we're not killing off our neurons nilly-willy. The blood-brain barrier acts as a barrier between the blood and the brain. It's exactly what it sounds like. The major component of the blood-brain barrier would be tight junctions between vascular epithelial cells. And these tight junctions are going to prevent diffusion between the cells. So anything floating around in blood, as long as it's bigger than about 400 Daltons and fairly hydrophilic, won't freely move into our brain. Now things like oxygen, carbon dioxide, those are small, and those are nonpolar. They're going to freely move across. But larger things, such as blood cells themselves, proteins, carbohydrates, those are going to be stuck in the blood until we transport them over. The blood-brain barrier is an important protective layer that prevents our neurons from being damaged. When there's injury, though, the blood-brain barrier can become permeabilized. Part of this is to allow immune cell entry, and some of this can also just be the result of neurodegenerative processes. When we have permeabilization of the blood-brain barrier, this can lead to nervous system damage. So we want to see this intact. Now, just because something can't freely move across the blood-brain barrier doesn't mean it can't enter the brain. We have a few ways to get in. So if it's something like glucose or insulin, something that we want to bring into the brain, we have transporter proteins for that. Glucose will be transported by glucose transporters. You find them on the vascular epithelia themselves, you find them on astrocytes surrounding them, and you find them on neurons. So, if we look in the top panel there, this is just reminding us that astrocytes surround blood vessels. In the bottom panel, astrocytes are shown in green and a neuron is shown in red. This is just showing us that a bunch of different astrocytes are going to help feed that neuron. When neurons are active, we get an increase in blood flow. That allows us to pull out oxygen and glucose and feed that neuron that was just active so it can create more ATP. We'll talk more about this in Lecture 5. But neurons and astrocytes are going to communicate with the vasculature to regulate their size. Astrocytes are a necessary middleman to keep neurons protected from the stuff in blood. So they're going to pick up things like glucose and transport them on over to neurons in an activity-dependent manner. More on that later. Now just a bit on evolution. So we see an increase in the complexity of organisms and their behavior because of an increase in the complexity of our nervous systems. So not every animal has a nervous system. Sponges, for example, have what we call independent effectors. So these would be cells that have both sensory and motor function. There's no connection between them and each other. They act independently. Cnidarians, such as jellyfish, have a nerve net. So now we actually have a nervous system. We don't really have a true brain. There's not a whole lot of encephalization going on. You'll see a little of this whenever we move into worms. So here we're going to have two nerve cords, one running along the ventral portion, one on the dorsal portion. There's some ganglia. They're going to help control different parts of the body that are connected by these cords. And there's a nerve ring that can act as the brain there. We're going to go from two cords to one. Whenever we move into insects, it's going to be ventral rather than dorsal like us. And we'll have a brain. Moving on to vertebrates, we have, of course, a true brain because it's actually encased in a skull. It's fairly complicated. We have a central nervous system, spinal cord. But the brain is really the star here. That's what's changed the most throughout evolution. Spinal cords, no matter what species you're talking about, they're going to have sensory and motor pathways in basically similar areas. They're going to have reflexes that are carried out within the spinal cord. So the processing is very similar. You'll see automatic coordination of bilateral alternating movements. The big change comes in the neocortex. That's the part that has expanded most dramatically throughout evolution. And that's where we think the magic happens. Of course, the magic of conscious perception, thoughts, plans, memories, behaviors. So all the things that make us us probably are going to be created in the cerebral cortex. Now, on a molecular and cellular level, what's changed, well, are many things. What I want to highlight here would be an increase in the abundance and diversity of inhibitory neurons. So, the GABAergic neurons within our cortex are present in much higher percentages in primates, like us, compared to rodents. So you see 15% of the cortical neurons being GABAergic in rodents, move that up to about a quarter of them in primates. And you see greater diversity in terms of their size, shape, the types of neurons that they target, their firing patterns, the types of proteins that they express. So we think that we have many additional classes of inhibitory neurons as you move up the evolutionary ladder. And the way that we characterize these neurons is shown here in this slide. So there's a few things we look at. What types of cells are they targeting? So if they target an excitatory neuron, then they're truly inhibitory. If they're targeting an inhibitory neuron, then they're kind of disinhibitory because they're inhibiting inhibition. So an important marker that you'll see is parvalbumin. This comes up pretty frequently. It's a commonly expressed calcium binding protein. Not every neuron expresses it, so you'll hear like PV positive interneurons. Those would be parvalbumin positive. But by looking at what genes they express, we can then isolate different classes. For example, if you express a certain type of neuropeptide, for example, well, that's going to inform us with your function, because different neuropeptides have different receptors, which will then have a different effect. So some neuropeptides can be excitatory, while others are inhibitory. So that can help distinguish different classes of inhibitory neurons. And then we can just patch clamp them, see what their firing looks like. Is it steady? Do they adapt? Meaning, does it change over time? Do they pacemake on their own? Do we see bursting? Do we see irregular firing? So there are many ways that you can characterize this diversity of GABAergic neurons that have come up. And we use the same things for excitatory neurons as well. In addition to the changes in neurons, we see a change in glia. And specifically what I'm talking about here would be astrocytes, or their an analogous cell in other species. So, when you look at the astrocyte to neuron ratio throughout evolution, you can see that this changes quite a bit. So, in the lower organisms, such as the worm, for example, or in rodents, we see a whole lot more neurons compared to astrocytes. But whenever you move up the evolutionary ladder, you see an increase in the number of astrocytes per neuron. Now, of course, this varies wildly from brain region to brain region, and this is a very hard number to just put a single value to, but the general trend is pretty well true. You see an increase in the density of astrocytes in the nervous system. Those astrocytes that are there are also much larger. So comparing a mouse and human astrocyte, you can see human astrocytes much bigger. Neurons are a little bit bigger, but they have not expanded to the same degree as astrocytes. So astrocytes are, I don't know, about tripled in diameter, whereas the cell bodies of neurons have a 50% increase. You can see an increase in the number of branches that are coming off, and of course the volume that they occupy. As that increases, the number of synapses that they target increases, so likely the complexity of their function has increased as well. So astrocytes are more complex and doing additional things in higher organisms. That's the idea. There's a lot that we still don't know in that realm. I don't want to increase the complexity of this talk anymore, so I'm going to end it here. If you have any questions, fill out the questions box so I can address those before we meet with each other. And if you have no questions, great. I'll see you later.